Good evening, everyone. Today, in our cycle of seminars and conferences, our very welcoming guest is Dr. Gabriel Abraham Cardoso Ugarte. He got his bachelor degree in gastronomy from the Universidad Autónoma del Estado de Mexico and his master and doctorate degrees in food science from the Universidad de las Américas, Puebla. Nowadays, he is a professor in the School of Gastronomy of the Universidad Popular Autónoma del Estado de Puebla and researcher in the same university whose lines of research focus on functional food formulation, fermentation, probiotics, and emerging technologies in food processing. Thank you, Gabriel, for accepting our invitation. And I give you the floor. And at the end of the conference, I think that some people will question your conference. Thank you.
Very good, Antonella. Thank you very much for this warm presentation. And yeah, good morning here in Mexico, and good evening in Italy. I also have to, before starting, I also have to thank to Professor Paolo Corbo, who sent us the invitation to the Faculty of Gastronomy of UPAEP through the director, Dr. Monique Orduña Sosa. So she sent us an invitation to take part of these conferences, and I said us because there's another colleague that will be taking part of these conferences, Professor Paula Vera. She's going to be giving another lecture, I think in April. I don't remember, in upcoming days. But yeah, thank you very much. I feel so happy to be here. And today the lecture that I prepared for you for Eunice has this name, exploring the functional potential of Pataxte, Theobroma bicolor. And yeah, well, before starting, I think that when you listen this word, let me check the pointer. When you listen to this word Pataxte, maybe you may think, maybe you are not familiarized to this word. It sounds weird for you. The same for me. The first time that I heard this word, I had no idea what was this about. When I see this fruit, this is the shape, the external shape, the outer part of the fruit. When I saw this one, when I saw this picture, I didn't have idea what was I going to find inside the fruit. So yeah, the name, I was also not familiarized with the name, but then when I saw the scientific name of this fruit, I started thinking more about what I was going to find, what was the fruit about. So maybe as you all people may know, the name Theobroma of the scientific name, maybe, I am sure that you are familiarized to this word, the Theobroma. This is the same family, the same scientific name of the cacao. The difference is that is Theobroma cacao. In this case, it is Theobroma bicolor. So when you listen to this word, maybe you can think about what it is going to be about the fruit, okay? So yeah, my name is Gabriel Cardoso, and I am a professor and researcher at Universidad Popular Autónoma del Estado de Puebla, UPAEP, here in Mexico, in Puebla. So I will start the presentation, the lecture. Before starting this, I have to start contextualizing the topic, okay? As I told you, well, this picture is not about Pataxte. This picture is, set my clock. I'm sorry. This picture is actually about cacao. Maybe all of you know this fruit. This is the fruit of cacao after harvesting, and inside we have the seeds and we have the pulp of the cacao. So I only put this picture only to make sure and to contextualize that, we are talking about a fruit that is part of the same family of the cacao. So this is all the scientific classification of the Theobroma bicolor. It is not necessary to read all of this, but I think that the most important part here is the genre that is the same as I told you as cacao. The genre is Theobroma, and I think these arrows are not okay. But yeah, this arrow belongs to Theobroma, and this one, the name Theobroma bicolor belongs to this Macambo or Pataxte. This is the name that we give to the fruit that I am going to be talking about. Macambo is more common in South America, and Pataxte is more common here in Mexico and in Central America. I am sure all of you know this fruit, the Theobroma cacao. You all may know that all over the world the seed is exploited and used to produce cacao, to produce chocolate. Okay, so that is why it's so important, this fruit, all over the world. So let's continue contextualizing this topic. So this picture, it is actually a picture of Theobroma bicolor. This is a picture of Pataxte. This is the tree where it grows. In this part, I don't know if you can see my pointer. This is the fruit hanging off from the tree, and this is a zoom of the picture of the fruit. So it possesses a similar shape as cacao. It is more rounded, but in the external part, its shell is more kind of rough or wrinkled, you know? So this is a picture when the fruit is on the rip, so it is green. Whenever it ripens, the external shell turns yellow, as you can see in this picture. And in this another picture, you can see the external shell removed in the upper part, and inside we have the flesh. Well, actually, the flesh is yellow, it's an intense yellow. The color is so similar to mango, and inside this flesh, we have the seeds, okay? The seeds are so different than cacao. Maybe you can remember if you have seen a seed of cacao, it is a deep dark brown seed, colored seed. In this case, the color is so white, so kind of creamy, okay? And the taste of the seed is so similar to a nut, I don't know, to pecans, to I don't know, the family of the nut. It is a nutty flavor, okay? And the flesh, the pulp, as I told you, is so fresh, is so yellow. The flavor is so intense, and the aromas are so similar to mango and to banana, okay? So the first time that you open this fruit, you feel a lot of aromas and flavors whenever you eat it. So the etymology of the name of this product is Theobroma. As I told you, the family is Theobroma, that derives from the Greek food of the gods. Okay, here in the ancient times, the Theobroma food was considered as the food of the gods. The Pataxte grows in humid forest and rainforest soils, mainly in tropical weather. So here in Mexico, we have several states, several places where we have this tropical weather, mainly in the states of Chiapas, of Oaxaca, maybe in some places in Guerrero, and also in some places here in the state of Puebla. The average weight of the fruit is 400 grams. As I told you, it has an oval shape, rough shell, and it's yellow whenever it ripens. It has an average of 40 seeds. As I told you, these seeds that are so hard and they possess a nutty flavor. And the seeds are surrounded by a strong yellow flesh with a sweet and intense aroma and flavor. This is important to mention that the consumption of the pataxte, the whole pataxte, the whole fruit, is scarce here in Mexico. Okay, many people does not know this fruit, so therefore the consumption is low, is scarce. But the people that used to consume this fruit uses to eat it in fermented drinks, and the seed is used to produce marzipan and nougat. So because of the similar texture and attributes of the seed that, as I told you, is similar to a nut. So after contextualizing what the fruit is, Maybe you may be asking why to explore Pataxte, why to work with Pataxte. So in Mexico, Theobroma bicolor is far from being domesticated as Theobroma cacao. As I told you, all over the world know Theobroma cacao, and the cacao is super exploited and super produced, and many people is trying to produce and to harvest cacao in order to sell it or to produce cacao or for producing chocolate. In contrast, Theobroma bicolor is so far from being domesticated because people does not even know it. So there's no comparison regarding the production of both fruits. Okay. One of the most important things, the most important features that I take into consideration when I decided to work with this fruit is that the people consider the Theobroma bicolor de Pataxte as a defective cacao, since the physicochemical attributes are so distant, so different than cacao, than Theobroma cacao. People think that it's defective because you cannot make chocolate with this. But yeah, it is defective for that purposes, but you can take advantage of the fruit in order to make different things. So there's when I saw me and a group of researchers in the faculty of at UPAEP, gastronomy, biotechnology, and nutritionists, all of these people saw an opportunity area of researching and looking for more information regarding the Pataxte. The pulp is scarcely used. It is used only, or in most of the cases, as fertilizer for the next sowing, and to a lesser extent, it is used in the preparation of jams, of nectars, and in a lesser extent as fresh fruit. So it is underutilized. Okay, this fruit is not exploited in Mexico, and that is the reason why this name. We maybe you know Beatrice Guzzi, that is I think is a student at UNISC. She is part of the magazine The New Gastronom, and she wrote an article that she named Cacao's Unlucky Cousin. Okay, and yeah, it is an unlucky cousin because it is part of the same family of the Theobroma cacao, but it possesses different attributes that make it unlucky because people is not using it. But well, this name is completely authored by Beatrice Guzzi. The article is published in the New Gastronom magazine that you guys may know, and yeah, all the name is hers. And then also in here in the name of the article, she wrote Pataxte, a superfood in the making, because we noticed that there's a lot of attributes and we have a functional potential inside the Pataxte. So those were the reasons why we decided to explore Pataxte. Okay. So the first question that we made when we had the fruit in our hands was this one. Is there prebiotic and antioxidant activity in the seeds and pulp of the Pataxte fruit? Why prebiotic? Because as I told you, when you see the flesh, it is too similar to mango, the flesh, okay? And to a fruit that is, I don't remember the translation in English, is guanábana in Spanish, so it is so fibrous. So when you see a fibrous fruit, a fibrous food, you can tell only by seeing it that it can maybe possess prebiotic activity. Let's remember that the prebiotics are the food for the probiotics, for the beneficial microorganisms. So we said, okay, maybe this flesh possesses prebiotic activity. Let's find out. And antioxidant activity. Also, we said, okay, so the Pataxte is the cousin of the cacao. They are similar. They may possess similarities in the physicochemical attributes. So yeah, we all know that cacao is an excellent antioxidant and it helps in the heart disease. So we said maybe Pataxte will also have this attribute. So those are the reasons why they say, okay, maybe we will find prebiotic and antioxidant activity, and we started looking for this. So in this research, in the research that was made by a student of the master degree in biotechnology at UPAEP, we started doing this analysis. We made these 5 analyses in order to explore the Pataxte. The first one, we made the proximal composition of the pulp and the seed. all of these analyses were made, well, except this one, the fatty acids profile, were made for the seed and for the pulp, okay? So we started determining the proximal composition. We looked for the presence of inulin. We also assessed the prebiotic activity of both. We also measured the antioxidant capacity, and we made a phytochemical analysis. And finally, as I told you, we made a fatty acids profile for only for the seeds because of the nature. Okay, so I will be describing briefly all of these analyses. So the first one, I apologize because of these names. Okay. Yeah, Antonella, thank you very much. Yeah, that is the link of Beatrice Guzzi. And well, in this article, she also interviewed a local producer of Pataxte here in Mexico. She also interviewed to the student that developed the experimental part of this work. And yeah, it's a more complete article. Okay. So as I was telling to you, I apologize because of this. In the normal PowerPoint, they were not translated. I think something wrong happened here, but well, I think it is clear enough. So the first results that I'm going to share with you is the proximal analysis. Remember that the proximal analysis is when we determine what is all the fruit composed of. So we determine the amount of carbohydrates. I am sorry because of this in Spanish. Carbohydrates, protein, lipids, ash, and moisture. Okay, so in the case of the seed, what is the most important or the most relevant information here is the amount of lipids. As you can tell, 30.73% of the seed was composed or is composed of lipids. So it is confirming that it's so similar to nuts. It may only may belong to the family of the nuts because all of this lipid content. Okay, of course the moisture content is so low, and the carbohydrate content is also high, and the protein content is also high. And on the other hand, we have the proximal analysis of the pulp. As you can tell, the difference is huge. Okay, the moisture, of course, is enormously higher than the seeds. We have here 37.25% of moisture. Then we have a lot of carbohydrates. We have 44.30% of carbohydrates. So in this initial stage, we confirmed that we have a lot of fiber in the pulp. The fiber that we saw in the pulp, it is being confirmed here in this amount of carbohydrates. And we also have a reduced amount of proteins, and of course a reduced percentage of ashes, and a so low, so tiny amount of lipids, only 2.88%. So the most important findings in this first stage of the research were these ones: the high amounts of lipids in the seed and the high amounts of carbohydrates in the pulp. Okay, so when we saw these results, we started thinking on the next steps. What are we going to do? What are we going to make to take advantage of all of these results, all of this information? So, yeah, as I told you, when we saw that we have a lot of amounts of carbohydrates, we said, okay, maybe in all of these carbohydrates, and as I told you, when we saw the pulp, we said all these carbohydrates might have prebiotic activity, prebiotic structures. As I told you, and as you all guys may know, the prebiotics are the food for the probiotics. So I put this picture because it's so graphic and so specific. The probiotics are actually eating the prebiotics in order to grow and to reproduce. Let's remember that the probiotics are those beneficial microorganisms, also known as lactic acid bacteria. Most of the probiotic microorganisms are part of this family, the lactic acid bacteria. So in order for the probiotics to grow, to reproduce, and to be to colonize our gut, they need food. Okay, and one of the most important food for the probiotics, of course, are the prebiotics. So we said, okay, maybe we can find prebiotic structures in the carbohydrate, in the flesh, in the pulp of the Pataxte. So we thought about one of the most important and known prebiotics in the world, the most successful prebiotics in the world, that is inulin. So we said, okay, we have to base in the structure of the inulin. So let's find out if we have inulin in the flesh or something similar to inulin that can work and that can act as a prebiotic. So the next step we made was the determination of inulin. So we're having problems here. Let me see. There is no images here. Give me 1 second, please. I will change the slides because these figures are so important. Please hold on. I will try to make this fast. I have the slides opened here, the PowerPoint document, so it should be fast.
If you
Please
need
let me
some
know.
help.
Yeah. Excuse me, Antonella?
No, if you need some help, I'm here.
Thank you very much. Yeah, I am familiarized with this software. It's the same that we use at the university. So I have uploaded the another slides. Can you see them? Yeah, are the slides visible for all of you?
Yes, yes, it's not in the, yes, it's not a
Thank you.
presentation. We see your PowerPoint, but we can see your slide without problems.
Thank you, Pau. Gracias, Pau. Okay, so I will try to set the presentation. Okay, I think this way is okay. I will make them, I will zoom here for doing the slides more visible. So, okay, as I was telling to you, after we decided to explore if we had some inulin in the pulp, we made an analysis of the structure, the main structures that the seed and the pulp possessed. We applied this technology, this methodology, the infrared spectroscopy, coupled to Fourier transformate. These are the initials of this methodology. So we obtained these spectrograms, okay? These spectrograms show us the structures that the pulp and the seed possessed, okay? So also we made the same analysis to the inulin in order to know if this spectrogram was similar to, I mean, if one of these spectrograms, the seed of the pulp, were similar to this one. And as you can tell, these two spectrograms, the ones of inulin and pulp, are so similar. So the first thing that we noticed was that the three samples, the inulin, the seed, and the pulp, showed this peak in here that shows the presence of water. So there's no more information in there. The important information here and the important similarities that we found are here. In this point, in this circle, this another circle, and in this peak, there is no circle in here, but it should be here. And here, we found similarities. So we said, okay, the structure of the inulin and the pulp is so similar. So maybe we are having inulin or similar structures to the inulin in the pulp. And the most important one is this one, this peak that is shared by the inulin and the pulp. It shows that we have there a lot of amounts of carbohydrates, oligosaccharides, that mainly act as prebiotics. So we said, great, maybe we don't have inulin because this methodology, this technique does not allow us to confirm that we have inulin, but we can see and we can confirm that we have a similar structure than inulin in the pulp. So those were good news for us, and we continue the work by exploring the prebiotic activity of the pulp. Okay. So these are the first results of the prebiotic activity of the pulp. Also, we tested the prebiotic activity of the seeds. How we made this? We put some lactic acid bacteria to grow in mediums that were combining an extract of the pulp and a combination of mineral salts that the microorganisms need also to grow. So in this case, we can see that we have 80% of extract and 20% of the mineral solution. In this case, 70% and 30% of the mineral solution, okay? So we tested 3 microorganisms, 3 lactic acid bacteria. We tested Lactobacillus brevis, Lactobacillus paracasei, and Bacillus mojavensis. This specific microorganisms is a microorganism. This one, the Bacillus mojavensis, was isolated from the agave, okay, that is utilized for the production of pulque, a fermented beverage here in Mexico. So as we can see in the case of the pulp, here we have the specific data. They are not necessary to read all of them. I think it is more important to see in this picture that the best growth in the pulp was reported by Lactobacillus brevis and Lactobacillus paracasei. So the same letter, the A, and A here, shows that there was not a significant difference between these 4 microorganisms, but we can see difference regarding the Bacillus mojavensis. So the pulp in both percentages, in both ratios, was effective for the growth of these microorganisms, okay, with no significant difference, okay? So it's the same, it was the same to use this ratio or this ratio in both cases. The Lactobacillus brevis and paracasei growth in the same rate, at the same rate, okay? And the lowest growth, as you can see here, was reported by Bacillus mojavensis, and here are the specific data, okay? So we found prebiotic activity in the pulp. So the pulp was effective for the growth of the Lactobacillus brevis and Lactobacillus paracasei. And here we have, I wanted to show you this picture of the growth. You can see the difference, okay? For example, in here we have 4 dilutions, okay? In this first quadrant, this is the picture of Bacillus mojavensis. In this first quadrant, that is the lowest dilution, you can see only 1 colony here, 1 here. This is the same, this is a triplicate. So we have only 1 colony, 1 colony, and 1 colony in the case of Bacillus mojavensis. In the case of Lactobacillus brevis, we have 1 here, we have several more here, like 5 to 6, and 2 of them. So we can visually determine and see that Lactobacillus brevis grows better than Bacillus mojavensis. And in the highest dilution, that is this one, this is so uncountable colonies. So in order to determine, in order to count these plates, we have to find between 25 to 250 colonies. In this case, it is uncountable. So we can see that the growth was more effective in Lactobacillus brevis than in Bacillus mojavensis. So we are only confirming the information that we saw here, okay? So the next slide is about the prebiotic activity of the seed, okay? So these results were so interesting because we were not expecting that the seed possessed prebiotic activity because we didn't see the presence of inulin, of any oligosaccharide in the seed. However, the seed also reported a good prebiotic activity and a good growth of the lactic acid bacteria. And the behavior was almost the same, okay? As you can see, Lactobacillus brevis and Lactobacillus paracasei growth in the same rate, okay, with no significant difference between these 4 bars. And the same Bacillus mojavensis was not, the growth was not so good as the one of Lactobacillus paracasei. So the same phenomena was reported in here. And there's another picture, is quite different than the one that I showed you before. As you can see, the difference is more notorious here in of the seed in the first quadrant. That is the first dilution, that is here, that I am pointing here. We have no growth. In the first dilution, there's no growth. In the case of Lactobacillus brevis, we actually have growth in this first dilution. We have 5 colonies here, the same here, and the same here. So it is also so clear that also the seed was a good prebiotic. But the most interesting part here is that the prebiotic activity comes from different structures. As I told you, the prebiotic activity of the pulp was mainly attributed to the presence of inulin. It was not inulin per se, but the structure was so similar. So the prebiotic activity of the pulp was because of the presence of all these oligosaccharides, carbohydrates of medium chain. In the case of the seed, this prebiotic activity might be attributed to phenolic compounds. The phenolic compounds are also responsible for the growth of microorganisms. They are also considered as prebiotic. We are only hypothesizing this affirmation. We have to make more research in order to confirm that the phenolic compounds that are present on the seed are the responsibles of this prebiotic activity. Okay, so then we finished with the prebiotic activity. We confirmed that both the seed and the pulp possessed prebiotic activity. Then we moved to the determination of the antioxidant activity of our samples of the seed and the pulp. So the first result that we found, the first analysis that we made was the qualitative test. Okay, in this picture you can see 2 plates that were rinsed with DPPH reactant. Okay, so the nature of these plates were violet. Okay, so all the plate that is full of the reactant, okay, is violet. So the first step is to put a part of the ethanolic extract, a drop of the ethanolic extracts, and if we notice a change of color from violet to yellow, we can see that we have antioxidant activity. So this first qualitative test is important only for saying yes or not. We cannot quantify how powerful the antioxidant activity is. Okay, we can only say, okay, yes, it has or it does not have antioxidant activity. And we can see in both pictures that, yeah, the seed ethanolic extract and the pulp ethanolic extract possessed antioxidant activity because of the change of the color. Okay, in this case, this picture is more clear than this one, but only for the light of the picture, but the activity is present. Okay, so how can we tell that we have activity, antioxidant activity? Because the DPPH violet color turns yellow as the oxidizer radical, this radical DPPH, gets reduced. Okay, so this was the first one, the qualitative test. Then we said, okay, we have antioxidant activity, let's measure this one. And then we made these calculations and these determinations. Okay, we also use the reactant DPPH. Okay, it's one of the most utilized radicals in order to measure the antioxidant activity. So what the extracts were making were like catching, like chelating the DPPH radical. So you can see here also the change of colors. Okay, these are some samples. This is the DPPH. Then we added droplets of the extract, and the color started to change. Okay, to reach this color. So in order to quantify the antioxidant activity, we measured the absorbance of these tubes, of these solutions. Okay, and with the absorbances, we were able to make this calculation of the antioxidant activity of the pulp and of the seed. So in this graph, in this picture, you can see the antioxidant activity of 3 samples. You can see on gray color the antioxidant activity of the pulp, on orange color the antioxidant activity of the seed, and in blue color the antioxidant activity of ascorbic acid that was used as control. Is one of the most powerful antioxidants. So if you are doing an antioxidant activity test, you have to compare with a control. in order to determine how effective is your antioxidant compared to a powerful antioxidant, in this case, the ascorbic acid. So I think it is so clear that the seed possessed a very good antioxidant activity, so effective, so powerful, even higher than the control, than the ascorbic acid, that, as I told you, is known for its powerful antioxidant activity. So this is one of the most important results that we obtained in this research. And as I told you since the beginning, we said, okay, it is from the same family than the seed, so it might possess antioxidant activity. And yes, it has. And it is even higher than ascorbic acid. And well, the pulp, we only made this measurement only to know, but yeah, it is so low, and it is not as important as the seed is. So then we calculated this coefficient, the IC50, that it expresses the amount of the extract that we need to reduce the 50% of the radical of the DPPH. And so in practical terms, as this amount is lower, is the more effective, the most effective. Okay, so as you can see, we only need 0.45 micrograms per milliliter of the extract in order to reduce 50% of the DPPH. In the case of the pulp, we need 10, and in the case of the control, we need more, 0.75. Okay, so it is only to confirm the powerful and effective antioxidant activity of the seed. And then we are about to finish. Then we decided to make this phytochemical analysis in order to know what is inside the seed and the pulp, and which are those metabolites that are responsible of the activity, the antioxidant activity. So as you can see here, we determined the presence of alkaloids, saponins, triterpenoids, tannins, flavonoids, cyanogenic glycosides, cardiotonic glycosides, and sesquiterpenic lactones. And the most important findings in this part, well, as you can see here, these symbols means abundant, then moderate, and then scarce. So the most important part, the most important findings in this part of the research were here the presence of alkaloids. There's a lot of alkaloids in the seeds. Okay, they are also responsible, might be responsible of the antioxidant activity of the seeds. The tannins, okay, this abundance of tannins that present in the seed, the tannins are so powerful antioxidants. They are also present in wine, so also in chocolate. So we are also hypothesizing that the powerful antioxidant activity in the seed is given by the presence of these tannins. And as you can tell, there are no tannins in the pulp. And finally, also a very important finding here is the absence of cyanogenic glycosides, cyanide, venom. Okay, so of course that it is desirable to not have cyanide inside any food. Okay, so this phytochemical analysis also gave us very good information. And finally, as I told you, when we obtained the results of the proximal analysis of the pulp and the seed, we saw that the seed was so rich in lipids. So we said, okay, let's find out what kinds of fatty acids does the seed have. Okay, so this is the spectrogram of the fatty acids profile of the seed, only of the seed. It was not necessary to make this analysis for the pulp because there's no lipids in there. So we find that the most abundant fatty acid was the palmitic acid, okay, 39.23%. Then we have also the second most abundant fatty acid was the oleic acid, 35.34%. And this one, the oleic acid and the linoleic acid, were so important. It was very good to find them because, as you can tell in its composition, they possess unsaturation. They are unsaturated fatty acids. As you may know, well, this is monounsaturated. It only has a double bond, and this has two double bonds. That means that this one is polyunsaturated. It's a linoleic acid. The linoleic acid actually is one of them essential fatty acids. Okay, the essential fatty acids are those that we need, but that we don't produce, that the body does not produce. So we need it, so we have to get it from the diet, from the food. So in the case of the seed, it possesses one of the fatty acids. There are three fatty acids: linoleic or linolenic and arachidonic acids. So we have the linoleic. It was so good to find this fatty acid in here. And also this one, that is a monounsaturated fatty acids. They are so healthy. And this one, although it is not, it is a saturated fatty acid, we can take advantage of this palmitic acid in order to prepare products like marzipan, like nougat, or chocolate-like bars. Okay, because this abundance means that at room temperature, the lipid is semi-solid. Okay, so these were the final findings that we believe that were important to get from this fruit, the pulp and the seeds. Okay. So in order to finish with this lecture, I think it is time. We can say that we have high amounts of lipids in the seed. We found high amounts of carbohydrates both in the pulp and the seed. We had an inulin-like structure in the pulp. I repeat, inulin-like. It is not the same. We found a better lactic acid bacteria growth with the extract at 20% in the pulp and in the seed. We also found that there is a growth of Lactobacillus brevis and Lactobacillus paracasei with no significant difference, so the growth was similar or equal, statistically equal. We also see, and I think this is one of the most important findings of the research, that we have an effective antioxidant activity of the seed, and it was even higher than the control. We found high amounts of tannins in the seed, and we found the presence of unsaturated fatty acids. Okay, so these are the main findings, the final remarks of the presentation of the research. But we are continue working with this. So what is next? Which are the determinations that we need to make in the upcoming days whenever the labs are open again, whenever we can go back to university? We have to confirm the presence of inulin to different analyses. We want to quantify the concentration of tannins in the seed, since only a qualitative determination was carried out in this study. And finally, we are trying to make the formulation of a symbiotic functional beverage using seeds or Pataxte pulp. We were starting doing this functional beverage. We were doing a powdered beverage using spray drying. We were trying to encapsulate microorganisms in there and using the Pataxte pulp in order to make a powdered instant drink. But the pandemic appeared, and so the work was interrupted. We are hoping to take this work whenever we can go back to the lab. Okay. So finally, special thanks to these people, Azarel Castillo, who were the student that developed all the experimental work that is the hardest work in the research. Also to Drs. Beatriz Perez and Doctora Saidanelli, that were part of the advising, they were advisors of the Azarel Castillo. And finally, special thanks to Dr. Monica Orduña, the director of the Faculty of Gastronomy, the person who presented us the Pataxte, and also who opened the doors of the labs in order to make some preparation and some formulations of the Pataxte. All of them part of UPAEP. And that's it for today. That's it. I think we are on time, just on time. Thanks for your attention, and may you have any question, please let me know. And also you can find me in this email, gabriel.cardoso@upaep.mx, okay? So I will be glad to answer your questions if you have any. Alexandra, I can see a question from Alexandra. How can we keep up with the progress of this project? Well, I will try to be in touch also with Aaron Gomez and Beatrice Guzzi, who wrote the article in the magazine of the New Gastronome. So if we have new findings, new information, you have to be sure that we are going to share with them, hoping they can share with you, Alexandra. Thank you, all of you, Khalid, Kiara, Semir, Alexandra. Thank all of you for your attention.
Aaron is working in our communication office, if you need to find him. Beatrice was a student, but now she's an alumni. And
Okay.
now I don't know where Beatrice is. I know very well this student because we collaborate also for other projects, but now I don't know where. I will look to her because it will be very glad to have her notizie, news. But Aaron is always at the university. I have no question because I teach medieval history. And then I can say that your presentation was very clear, but I need also to read the article because it's a little difficult for me. It's another
different topics, different areas.
Yes, very different topics. But thank you because it was very interesting. But there is, I see a question of Khalid.
Yeah. Out of the pulp and the seed, which one has the higher potential to become a widely used superfood? I don't know, Khalid. I would tell that the seed, maybe the seed, because of the presence of antioxidant. As you may know, nowadays the antioxidant ingredients, foods are so valorized. So maybe because of that, but also the prebiotic foods are also being so famous nowadays. So I think that both possess that potential. Personally, I would like, and we are actually trying, we are developing another research with the pulp. We are making a beverage, not a powdered one, the one that I thought I mentioned in the presentation. We are developing a normal beverage, a liquid beverage with the pulp because of the flavor. So I think it is easier to make the consumption of any food if it tastes good. So maybe I can see more potential in the pulp only because the flavor, the aroma, and the color is so attractive. I hope I answered your question, Khalid. Thank you for the question.
Yes, and we have to wait for the end of the pandemic period, I think.
Yeah.
Then we will do our research and we will find some other elements very interesting.
I think that so many researches were stopped suddenly, were interrupted suddenly for the pandemic. So yeah, it's been a year. So we hope we can go back as soon as possible to continue with this.
Yes, we hope so. Any other questions?
Are the cost of production high? Well, I don't have the specific amounts of production, but as I told you, most of the times it is a backyard plant and it grows along with the cacao. So I don't think that is so that the cost is so elevated. I think it is so cheap. It is that cheap that the people even can discard it, put it to the trash, and they don't use it. So I don't think the production is so high, Kiana. You're welcome.
Then, if we have no other questions, I think that you can go to your breakfast.
Yeah, I think
No,
it is time.
Alexandra.
Yeah, Alexandra.
Alexandra.
Yeah.
Sorry, I will leave you to your breakfast in a second. I just have a question. I'm currently a student at the University of Economic Sciences, and I just had a class the other day where we talked about, or today actually, about how some of these discoveries are being exploited in order to make money. Are you even thinking about this problem currently? Like at some point when you be able to make a nutritious drink out of it, do you think about it already?
Yeah, well, we are not looking for getting money. We are looking for the exploitation, as you tell, of this kind of food, and more this kind of food that is underutilized. As I told you, it has been being discarded, wasted. So if we give a potential, if we give an extra credit to this kind of foods, we are able to make potential functional drinks, functional foods, and we want to aid to promote the circular economy. You know, we are trying to avoid the people to discard and to trash this fruit, and we are giving them. We want to give them information of what all these have inside the fruit has inside, in order they to take advantage. Okay. Yes, we want to make the drink in order to produce a symbiotic beverage, okay? Because we are trying to put in the same beverage microorganisms, lactic acid bacteria, and prebiotics, okay? But the people can consume that can keep consuming the food per se as a fresh fruit, but people is not doing so. So the information we are generating is in order to make the people aware of all the functionalities that the fruit has.
Okay, I see. Thank you very much.
Thank you very much, Alexandra.
Thank you. Then it's breakfast time, I think, but I'm very nice to meet you, to have met you, and to have met you, I don't know. And I hope we met in person in Pollenzo as soon as possible.
Yes, I hope so. I hope to be there whenever it finishes, to know Pollenzo, to know you, to know Professor Paolo, and to know the city, to know the university. Thank you very much for the invitation. Yes. I was so happy to be here, and I hope this lecture was useful for your students.
Yes, I think so. Thank you. Thank you very much.
Thank you. Thank you very much, Antonella. Have a good night. Thank you, Alexandra. See you. Gracias, Pau.